Okay, you should be able to access this project file once I share it if you have a Benchling account. First, you're going to click on this icon where it says projects, and then go to CRISPR sequences. You'll see a file for blue colonies, white colonies, and the LAC-Z gene. First, click on either a white or blue colony, and select one of the files. All of these abbreviations found above the sequence are restriction enzyme sites. You can scroll through and try to identify whether or not this white colony sequence has the NOT1 restriction enzyme site. So you can find it here. And if you have trouble searching through all of these restriction enzymes, you can also go to the top and click on this search icon and search the sequence for the NOT1 restriction enzyme site, which is GCGG, CCGC. Okay, and it highlights the site. So that's what we expect to see with the white colonies, because the donor DNA sequence had a NOT1 restriction enzyme site. We don't expect to see that in the blue colonies. Next, I want you to perform a sequence alignment. So over on this right-hand side, you see all of these icons. Select the one that says alignments when you hover over it, and then click on create new alignment. The template sequence will be the white colony sequence that I already have in my window now, but I need to select the LAC-Z plasmid sequence to perform the alignment. Okay, so once you add that, click on Create Alignment, and it's going to compare the two sequences. So down at the bottom, you see gray and red regions. The gray regions are where there are matches. Red regions are deviations from the template sequence, or mismatches. It's very common to see mismatches at the ends of a sequence file, and also we expect to see lots of red after this, in this LAC-Z plasmid sequence, because it's not really the full plasmid sequence that I have here, it's just the LAC-Z gene alone. So the actual sequence is much shorter than the white colony sequence file, so that's why we see all this red at the end. So really we want to focus on this region close to 150 base pairs. So if you look down at the bottom, there's a scale to show you where you are in the DNA sequence. So 200 is around 200 base pairs, and I want to show this region where there's red. Okay, so this is somewhere in the middle of the LAC-Z gene. Now if we look at the alignment map here, wherever there is gray missing at this top bar, that means there isn't a match. So there's mismatches between the bases here. It'll also be highlighted in red for the LAC-Z plasmid sequence. Okay, and in fact, this is where the NOT1 site is. So that GCGG-CCGC is the NOT1 restriction site. So in the original sequence for the LAC-Z gene, it was CCCTTTCG, but several of these bases were mutated due to the insertion of that donor DNA during the repair process of the CRISPR gene editing. Okay, so this colony, we can confirm that it has that NOT1 restriction enzyme site. You can also mention whether or not there are additional mismatches. So we have this T to A conversion at this location. And to get the exact location of that sequence, down at the bottom it shows you the start. So that's at the 76 base in our template sequence. Next, you want to export it. Because I have my screen sort of zoomed in so that you can see better, some of these toolbars are hidden. Let me just zoom out, see if it comes back. There it is. Okay. So if you zoom in too far, this export button disappears. So just zoom out, go to export, and then you can select PDF screen capture. Hold on, that doesn't look right because I have a base highlighted, so it's only exporting that sequence. So just click on the sequence again, but make sure you don't actually drag a specific region. All right, PDF screen capture. Okay, so now it's showing, just boost that up. So that's what it'll look like when you select that screen capture. It's going to give you 3 pages. Really, I don't need all of those. I only need to see the first page. So download it and then save it. And you'll include either the whole full page, or you can crop it to just highlight that region of mismatches in your lab report. Okay, so you're going to perform that again. So go back to CRISPR sequences. You can do another white colony. So do 2 white colonies and 1 blue. And then just perform the same process all over again. So you're going to look for the NOT1 site, and then you're going to perform the sequence alignment. In your lab report, you want to discuss whether or not the NOT1 site was present. Were there any additional mismatches or mutations in the LAC-Z gene? So were there insertions or deletions or base pair changes? Was the blue colony LAC-Z sequence identical to the LAC-Z plasmid sequence? How do these results compare with your NOT1 restriction digest that you performed? So for instance, in your white colonies and blue colonies that you purified DNA from and then performed the NOT1 restriction enzyme digest, did you see linearization in your white colonies and a lack of that linearization in your blue colonies? And lastly, why does the change in the sequence induced by the CRISPR Cas12a gene editing cause the colonies to be white instead of blue? So those are some questions that you want to answer in your lab report. All right, guys, if you have any questions or want to walk through this with me, I'll be available this evening and most of the day tomorrow. Friday I have a lot of meetings, but I think after 2 p.m. I'm free. Okay.